All right, so next week is our first exam. Uh, you have a little bit of time for that because we have off on Monday and then we have class on Tuesday. It's really important to realize, even though I've got a picture of Einstein, you don't have to be a genius. That if you plan and spread out your studying, you're likely to be able to do pretty well on the exam if you can study a little bit each day, half hour, an hour, maybe an hour and a half each day from between now and the exam. You'll do pretty well. And so uh, the most effective studying are short sessions that are repeated over the course of a day and then over several days. Okay, that involves not doing just one thing, but several things: reading, taking notes, writing, practice questions, quizzing, things like that. It's really important to do that. It'll help you remember. Okay, so really the Boy Scouts, the motto of the Boy Scouts is be prepared. Spend a lot of time, choose active methods of studying, taking notes, making review sheets, quizzing, or working with a partner. These are all ways that are better than doing highlighting or just passive reading or turning pages. Plan multiple times to study each day and take breaks. Don't A 3 hour session is less effective than maybe 4 half-hour sessions or 5 15-minute sessions. Um, so, uh, what can you do to study? Well, you should certainly by now look at the chapter summaries. Hopefully, you've studied each chapter, but if it's at this point, it might be too late to do a lot of reading and studying each chapter, but you can review headings, you can review terms in bold, you can look at chapter summaries, you can look at your notes. Of course, whenever you start something new, the most important thing is vocabulary. And so, um, identifying words that have specific meanings in biology and knowing what those words mean is really important. All right, so this exam, we cover chapters 1 through 3. The um, major topics in chapter 1 include science and then a bunch of chemistry related to biology. For science, science is really a way of evaluating alternative ideas that measure the ideas against the evidence. Science has lots of rules. It's an open evaluation, and we don't consider something a theory until most of the other ideas have been proven wrong and there's been multiple chances for the predictions of that idea to be borne out true. So that's a really important thing. Peer review, controlled experiments, looking at what is a fair comparison group is really important. Okay, and then I mentioned core themes in biology. Here's kind of the list of them in chapter 1: life properties, organization of living systems, cell theory, DNA, evolution, homeostasis, and the relationship between structure and function. Okay, for properties of life, all living organisms are organized into cells. Cells are the smallest living unit. The cells are sensitive and respond to the environment. They're capable of growth and reproduction and development. They can pass on, when they reproduce, they'll pass on their traits, but those traits can be modified through evolution. All living organisms use energy, so have a chemical metabolism and maintain a stable internal environment. That's homeostasis. Okay, levels of organization goes from very small to very large. In this class, we're going to work on chemical organelles, and cells will be the main things. In the biology 2, then it's living organisms, populations, ecosystems, and things like that. Homeostasis is how living organisms regulate their internal environments. We'll talk about this a little bit. They detect change, they respond to the change to restore normal internal conditions. Usually this is through something called negative feedback. Structure and function relationships. In cell and molecular biology, what this usually means is that molecules are shaped to do specific jobs, and they have how they interact depends upon their shape and their charge, and often These most important molecules here are proteins, and their shape is determined by the amino acid sequence and the folding. All right, then we'll talk a little bit about natural selection, the evidence for evolution by natural selection, and we'll get to that. Living organisms pass on genes. The genes that are most common are the ones that help the organisms survive, and that's the theory of natural selection. There's lots of evidence that supports organisms have arisen by that. Okay, that's kind of chapter 1. Then in chapter 2, we did some basic chemistry, talked about elements and atoms. The periodic table just arranges the elements by the number of protons and in columns by their properties. This corresponds to the kind of the reactivity. All atoms have particles in them, protons, neutrons, and electrons. These particles, the protons have a positive charge, the electrons have a negative charge, and neutrons are neutral but have mass. The number of protons determines the type of the atom. The number of electrons determines how reactive it is. Chemical bonding occurs when elements participate in an exchange or sharing of electrons in their outer orbitals. These are the valence electrons. And there are certain numbers of electrons that are stable. Usually either a full orbital or an empty orbital is most stable. Sometimes atoms will lose or gain electrons to become ions. Charged ions, negative charged ions are attracted to positive charged ions. That's an ionic bond. Covalent bonds are sharing of electrons. Water is a covalently bonded molecule. Each oxygen atom shares a pair of electrons with each hydrogen atom in water. Okay. However, in this case, they're not shared equally. Electrons spend more time near oxygen, and so oxygen has a slight negative pole to it, and so it appears kind of negative. The negative region of one water molecule will be attracted to the positive region of another water molecule. This is called hydrogen bonding, and it's not really a chemical bond, it's just an attraction. But it gives water its unusual properties. It's what makes it a liquid over a wide range of temperature. It's why it has cohesion. It's why water is a good solvent for many substances and can absorb a lot of heat. Most organisms are mostly water. Okay, then in the next chapter, we talked about organic molecules, carbon and hydrogen bonds. These have lots of carbon and hydrogen bonds. Carbon can form 4 bonds with other atoms, and so it will usually share electrons because it's not super strongly attracted to electrons, and so it usually just shares them. And this allows it to make lots of complex structures because it can form rings and chains and things like that. These complex structures can be used for many things, for storing energy, for enzymes, for receptors, for building structures. Living organisms generally have 4 types of organic molecules, 4 broad classes: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates contain carbon, hydrogen, and oxygen in a very specific ratio. The simplest carbohydrates are simple sugars like glucose and fructose. Their chemical formula is C6H12O6. And these are good sources of energy. They're easily used by the body. And so you have some examples there. Large carbohydrates are just these small sugars linked together to form large molecules. That's a good way to store energy. Starch is a good example of that. But it also, if they're put together in a way that's hard to take apart, these can form structures like wood. So it's there. Lipids are the fats and oils. They contain lots of carbon and hydrogen, but not much oxygen. And so they tend to be very are not very water soluble. They would rather dissolve in oils. They they don't mix well with water. Um, the examples of lipid molecules are fatty acids or molecules that are made of fatty acids like triglycerides. Phospholipids are made of fatty acids, but they have a polar region that it likes water, and so they form membranes. And then steroid molecules are usually ring structures that regulate physiology. Okay, here I'm going to do DNA first. DNA is a double helix molecule that has a sugar phosphate backbone. Inside are the bases adenine, thymine, cytosine, and guanine, and these form complementary pairs. So adenine is always paired with thymine, cytosine is paired with guanine. The sequence of bases can be in any order because they're just hydrogen bonded together, and the strands can be separated, so the molecule can be copied into a new DNA molecule or into a piece of RNA that is then going to be used to make a protein. And so the sequence of bases serves as a code for making proteins. It codes for the amino acids in the protein. So each protein is made up of a chain of amino acids. Anywhere from 10 to 500 amino acids might be in a chain. All the amino acids contain an amino group. They all contain a carboxyl group. Those can form peptide bonds with each other, and any amino acid can bond with any amino acid. Once they're bonded in a chain, the chain will fold up to form complex structures. Okay, that structure is really important. It's what makes proteins really, really useful. DNA is usually converted to a protein through RNA intermediates. And so this is kind of one of the core things of molecular biology.